Bismillahirrahmanirrahim. Assalamu alaikum, dear student. Today the topic is the cell cycle. In order for an organism to grow, whether it is a single cell or multicellular, three events must take place. The cell mass must increase, there must be a duplication of the genetic material. Number third, a division process must occur so that each daughter cell receive an equal and identical complement of the genetic material. In eukaryote, this occurrence takes place in an ordered progression of events during the cell's lifespan or cell cycle. The initial event in the cell cycle is the growth and increase in a cell mass that occurs following cell division. This picture showing on the phases of the cell cycle. Now, there are four phases of the cell cycle. This is called the G1 phase, G for gap. For a mammalian taking 24 hours to complete the cell cycle in G1 phase. It lasts about 10 hours. G1 phase is devoted to the metabolic activities associated with the cell growth and preparation of a DNA replication. Following G1, DNA replication occurred. This is the synthesis or S phase, which lasts about 9 hours. During the S phase, the genetic material of each and every chromosome is replicated. Thus, each chromosome consists of two sister chromatids joined at the centromere. After the completion of DNA replication, the cell enters another growth phase called G2 phase. During this post-DNA replication phase, which typically lasts about 4 hours, preparation for mitotic cell division are taking place. And then the last phase, the M phase or division phase, is the final part of the cell cycle. The division process takes about 1 hour to complete. The two sister chromatids separate from each other, one going to each of the two daughter cells. The two daughter cells are genetically identical to each other and to the parent cell. Here you can see the cycle: G1 phase, synthesis phase, G2 phase, mitosis. Gap phase, we can say G1 phase is a gap phase, and this is DNA synthesis phase, which is also known as S phase, chromosomes duplication, and here G2 gap phase 2, cell with a duplicate chromosomes, and then chromosomes separation, mitosis, the cell division of the cytokinesis. The capacity of a cell to produce itself, perhaps the most fundamental property of life. There are two main processes by which eukaryotic cells divide: mitosis and meiosis. Mitotic cell division, which occurs virtually all eukaryotic cells, is the process by which single cells reproduce themselves and multicellular organisms grow. The key features of the mitotic cell division is that the daughter cells are identical to each other and to the parent cell. The parent and daughter cells are diploid, 2n in number. They contain two copies of each chromosomes. This is not true in meiotic cell division. The results of the meiotic cell division, it is the basis of a sexual reproduction in all higher plants and animals. It occurs during gamete formation in animals and spore formation in plants and fungi. The parent cell is diploid 2n, but following by the meiosis, the daughter cells, gamete or spores, remain haploid. They contain one copy of each chromosome. Mitotic division, mitotic cell division, was first described by Walter Fleming in 1879. It consists of a two interrelated process: mitosis, the division of the nucleus. The term refers to the thread-like appearance of the chromosomes, and the second is the cytokinesis, the division of the cytoplasm and other non-nuclear component of the cell. The process of mitosis is important for all living organisms, as in intracellular organisms. As in a unicellular organism such as protozoans, fungi, algae, it is the basis of asexual reproduction. A multicellular diploid organism begins its life as a unicellular zygote. The zygote and the resultant daughter cells divide mitotically, that results in the development and growth of the organisms. The adults, the mitotic activity can be witnessed during wound healing and cell replacement in certain tissues. The new daughter cells produced as a result of a mitotic cell division are usually one half the size of the parent cell. If you measure the quantitative of DNA, it will show the same as parent material. The amount of the genetic material is same. This shows that during mitosis, the process of cytoplasmic division is different from that of a division of the genetic material. The process of cytoplasmic division is termed as cytokinesis or cytoplasmic division. This process seems to involve partitioning of the cytoplasmic volume into equal parts, which are later on enclosed in a plasma membrane. The cytoplasmic organelle either is detached or raised from the existing membrane structure or synthesized de novo. De novo means from the beginning in the each cell. The divisions of the genetic material involve replication and accurate distribution in the daughter cells and is therefore more complex and is called nuclear division mitosis, that is karyokinesis. Event of mitosis phase. Here you can see MPPMAT, prophase, metaphase, anaphase, telophase. An interphase cell is a meristematic tissues differ from an interphase cell in a mature tissue. Meristematic cells of an apical region are spherical without a large central vacuole, and nucleus occupies a major portion of the cell volume. Mitochondria, dictosome, endoplasmic reticulum, and ribosomes are similar to those found in an interphase cell of a mature cell, leaf cell. The plastid lack chlorophyll and are larger than mitochondria and possess only a few paired membrane. Here you can see kinetochore, microtubules, centromere region, and these two collectively called as chromosomes. Each sister chromatid has its own kinetochore. Sister chromatids become attached by their kinetochore to microtubule from opposite pole. This picture showing the stage of mitosis, the first stage of mitosis prophase. Here you can see the condensed form of chromosomes, and then these chromosomes reduce to form in a visible form. The unit fibers found in interphase nuclei are not seen at the onset of the mitosis, and definite chromatic threads are seen instead of the start of the cell division. Almost one-third of the process of mitosis is completed during this stage in animal and lower in plants. The two pairs of a centriole migrate to opposite pole. Here you can see the centriole. Chromosomes assemble themselves in the middle position. The distinguishing event of metaphase stage is the migration of the centromeric region of each chromosome toward the equatorial plane. The equatorial plane is the middle line region of the cell, a line which runs perpendicular to the polarities established by a centriole and a spindle fiber. We come to the phase that is anaphase. Anaphase is the most shortest phase of the cell division. It takes 2.2 hours in an onion cell. But some critical and precise events take place during this stage. During this stage, the sister chromatids of each double chromosome separate from each other and migrates to opposite ends to the cell. To facilitate this process, the centromere divide and each chromatid has its own centromere and now termed as chromosomes. The movement of the chromosomes to the opposite pole is guided by the centromere. The centromere led by the way while the chromosomes migrate to the opposite pole. Now, overall, this process assures that each daughter cell will receive one of the duplicate set of the genetic material carried out by the chromosome. The last stage, that is telophase. Prophase is essentially reversed. The mitotic spindle is disintegrate. The chromosomes decondense, nuclear membrane reform around the genetic material to form two nuclei. Each has an identical copy of the genetic information. Nuclei reappear and interphase cellular function resume. Cytokinesis divide the cell into two daughter cells. Cytokinesis usually begins in the telophase and ends shortly thereafter. In an animal, a cleavage furrows develop, usually close to the vein and the metaphase plate walls. A microfilament actin ring contract due to interaction with the myosin molecules, forming a tipping furrows. Eventually, the ring closes enough for a spontaneous separation of the plasma membrane, resulting into two separate cells, but different in a cytokinesis. You will find different cytokinesis in a plant cell. In plant cells, a plate develops in the middle of the cell with the help of alpha glucuronic acid, which produced by Golgi complex. Vesicles that originated from the Golgi line up in the equatorial region. Vesicles fuse at more vesicles, grow outwards until reaching to the plasma membrane and thus separating the cells. The vessels contain material for making the primary cell wall and a middle. Cytoplasm and with it most organelles is usually distributed randomly but roughly equally between the daughter cells. Sometimes cell division is a highly regulated polar division that purposefully distributes some materials unequally.